Hello everyone, I'm Jean-Guillaume. So I'm a member of the Visibility and Global Affairs of a subcommittee of EMEUNET. I'm a rheumatologist at Lille University Hospital, and for the moment I'm doing a clinical research here in Oxford. So happy to join.
And also I'll add that there is Daniele Muro, who is the leader of the Visibility and Global Affairs Subcommittee, who is with us today. So thank you all for your contribution. And I'm also very happy to welcome all the members of AYR. So it's the Young Society of EULAR that who will be joining us today in this session. So we'll start first this session by briefly introducing both young societies, EMEUNET and yEFIS, and then we'll proceed with the presentation. So at the end of each presentation, there will be time for questions. So start writing your questions through the chat. So first, I have the pleasure to present Tuwe Gastrop. So Tuwe is a rheumatologist and associate professor in immunology and pharmacology. He obtained his medical degree from Aarhus University in Denmark and his PhD from Aarhus University and Stanford University. Tuwe is actually the current chair of EMEUNET, and he will now present an introduction of EMEUNET. Tuwe, the floor is yours.
Okay, thank you very much. Thank you to the organizers, and thank you, Christelle, for that very nice introduction. I'm very much not used to that. Okay, so who is EMEUNET? So structurally and organizationally, we are a EULAR committee. So you can find us on the official EULAR website, and we are in the communities committees. But what we really are is a platform to grow for young members of EULAR. And we are currently more than 3,000 members. And as you can see here, you can register on our website. Currently, there is a little glitch on our website, so maybe there is it's not possible. But usually it takes less than 2 minutes to register. And this is sort of the structure. So you see all the members, more than 3,000 members. The people that do the organization of the work is the committee. So that's 9 people. And then there are the subcommittees. So each committee member leads a subcommittee. So really the people doing all the hard work are these 62 subcommittee members. And then in each of the EULAR countries, we have country liaisons. So we have someone to disseminate all our activities. This is maybe the most important slide today, because this is not only for rheumatologists. So you see that many, so more than 1/3 of the current subcommittees members, they're actually more into research than into rheumatology. So please join us on social media. We also have a podcast. You can find it on Spotify or wherever you listen. You can join our activities. So these are the webinars, but also at all the congresses, we have many different activities, and you will get the newsletter if you sign up to become a member. So that's it for me. Thank you very much.
Thank you very much, Tuwe, for this very nice introduction of EMEUNET and showing all the different activities. And I can see that we have many EMEUNET members also connected with us today. So also yEFIS member, you can also register and be part of EMEUNET. So now we'll present yEFIS. Rami, the floor is yours.
Thank you, Christelle. So the invitation is also you can register in Young yEFIS, of course. And I'm very happy to welcome our spokesperson, Dr. Myriam Nabhan. She did her PhD in France, actually at Paris-Saclay University, where she worked on protein aggregates and immunogenicity. And then she moved to Ireland and at University College Dublin, where now she works on cancer immunotherapy. She will be the only registered member who does not work on autoimmunity, but I hope that she will learn a lot and she will then dive into autoimmunity with us. So Myriam, the floor is yours.
Yeah. Thank you, Rami. I don't think I will be joining you guys, but I like my field. But I'm looking forward to hear about it and hear the application, and that could be also applied for cancer. So I'm the yEFIS spokesperson since this year. And as Christelle said, the yEFIS is the Young Immunologist Network of the European Federation of Immunological Societies, which is EFIS. Our goal is to represent all ECRs working in immunology in Europe and to bring visibility to these young immunologists and improve collaborations between young immunologist society, but also with societies of like other societies like EMEUNET. And also one of our goals is to connect young immunologists with more senior scientists, experienced scientists to shape the next generation of immunologists. And by young researchers, we mean anyone who has less than 10 years of experience, of working experience, so after their PhD. This is how yEFIS is structured. So we have the yEFIS leaderships that's formed by the spokesperson, vice spokesperson, and treasurer. We coordinate the yEFIS task force, which is composed of the our working groups and of the national societies. So we have different working groups that tackle topics like equity, diversity, career development, education, publishing, communication, and also organizing events for our network. We also have up to now 16 young national societies that have been created all over Europe and that represent the immunologists of each country in Europe. And we have, this has to be updated, we have more than 800 members registered in the yEFIS network, and we organize webinars like today. We organize also events for the Day of Immunology, for example, in April. We organize conferences, and we have subgroups, interest groups that tackle scientific topics. So as I mentioned, we have these different working groups and 16 young national societies that anyone can join as a member of the parent national society. yEFIS has been around for now 3 years, and one of the things that I want to highlight is the yEFIS first symposium that took place last year, or now 2 years ago, in Berlin. It was our first big event in person where we met more than 200 young immunologists, and now we're meeting again this September in Dublin at the yEFIS second symposium. We're also joining different conferences around the world to increase the visibility of yEFIS and also increase the visibility of young immunologists. So this is us at the first symposium, and hopefully we'll see some of you at the second symposium, which will be in Dublin this year in September. We'll have a satellite symposium that will be right before the European Congress of Immunology from the 1st to the 4th September. And registration is already open, so you can already register and submit your abstracts. And I'm gonna finish here by, you can scan this QR code and join us and become yEFIS members. And you can follow us on Twitter, LinkedIn, and Instagram to know more about what we do. Thank you.
Thank you very much, Miriam. And I think now we can dive into CAR T cells, and I will introduce our first speaker. I'm very happy to welcome Dr. Valeria Melendez-Mayorga from the University of Freiburg, Germany. And Valeria is very international. So she first did her bachelor degree in pharmaceutical chemistry and biology in Mexico City. Then she moved to Argentina for her PhD, where she did the master's degree in biomedical sciences before going back to Germany, where she performed her PhD focusing on the development of immunotherapies targeting glycans in cancer. And in 2022, she got her PhD. Then she joined the Professor Susanna Minget group at University of Freiburg to work on CAR T cells at a postdoc. And now she will tell us about new techniques to optimize these CAR T cells. And we are so happy to welcome you. So you can share your slides. And meanwhile, if you have any questions between the audience, please don't hesitate to put them in the chat, or you can ask them at the end of the talk.
Okay. So thank you very much for that very nice introduction. And hello, everyone. And I would also like to thank the organizers for inviting us to present our research work that is entitled Harnessing CD3 Diversity to Optimize CAR T Cells. Well, I will just give a very brief introduction because now I know the background of everyone here. So very basic, but okay, I would say that. Okay, the immune system is comprised of a very complex network of highly specialized cells responsible for identifying and destroying possible harms to our body. For example, viruses, bacteria, parasites, fungi, or even cancer cells. Cancer represents a major global health problem that affects social development and, most importantly, reduce people's life expectancy. Immunotherapy is one strategy that can help us to overcome the problem that we have with cancer. Immunotherapy redirects the immune system to recognize, attack, and eventually to kill cancer cells. Several immunotherapies are available, going from monoclonal antibodies, vaccines, oncolytic viruses to adoptive T-cell therapy. For us, it's particularly interesting T-cells view the critical role that they can play against cancer. T-cells can recognize and react to different self and foreigner antigens through the T-cell receptor. The TCR-CD3 is a multi-protein complex conformed by the TCR alpha-beta heterodimer, which is lacking the signaling domains, but together with the zeta homodimer, the CD3 delta, the CD3 epsilon, and the CD3 gamma, these intracellular domains constitute the signaling apparatus that is also responsible for the activation of T cells and the interaction with many other different molecules downstream. T cells may require either antigen processing or the presentation by antigen-presented cells to overcome or the presentation of the antigens by antigen-presented cells. So to overcome this limitation or this intermediate step of T cells were engineered with synthetic receptors that are chimeric antigen receptors, shortly CARs. CARs allow the direct recognition of surface antigens in cancer cells. CARs are chimeric antigen receptors that are formed by monoclonal antibodies and the signal machinery of T cells. So the first generation of CARs own only a single-chain variable fragment of an antibody that was linked through the transmembrane domain to the CD3 zeta chain of the QCR. Then, by the introduction of a co-stimulatory domain, the second generation arrived. And later on, more sophisticated constructs came and gave place to the third and even the fourth generation, adding more co-stimulatory domains or even equipping these CARs with the co-expression of cytokines. CARs can recognize an antigen on the cell surface of tumor cells, activate the T cells, and eventually kill and destroy these cancer cells. This immunotherapy has been successful, and actually it was approved in 2017, the first treatment towards CD19, and it was specifically directed to B-cell malignancies. Today, the treatment has a complete remission up to 86%, and even though this high rate of success, we can still see severe toxicities in patients, such as cytokine release and neurotoxicities. CARs are modular constructs in which we have a binding domain which commands the specificity of the CAR, and then this binding domain is linked to the co-stimulatory and activating domain through a hinge and transmembrane domain. Within the intracellular signaling domain, we have the co-stimulatory molecule and the activation molecule. Among the activation molecules, so far, the only molecule that has been used is CD3 zeta from the TCR. However, the TCR also has other intracellular domains, being gamma, delta, and epsilon. different motifs that are present in each of these subunits may play relevant roles in the CAR context. Today, the functionality of each of these intracellular domains are still missing. We have then engineered new CD3 CARs, each harboring one of the CD3 intracellular domains. The BB epsilon, the BB gamma, the BB delta, and the BB zeta CAR. We have used a backbone of the FDA-approved CAR, that is a second-generation CAR, and we systematically engineer this CAR to create the BB epsilon CAR, BB gamma delta, and BB gamma delta, and the zeta that was used as a reference in this work. Then, isolated human T cells from healthy donors were transfused with lentiviral cells that encoding the different CD3 CARs. These constructs were checked if they were coming to cell surface after a few days. And we could see that in the analysis, all the CARs were coming to cell surface in similar expression levels. We also include an empty vector that we will call here MAR. But next, we were wondering, and we wanted to challenge our novel CARs and check if they were able to kill CD19 positive cells. We use a luciferase-based assay in a target effector 1-to-1, and after 3 hours, we could see that all our CD3 CARs were able to kill these pre-leukemic cells in a similar way as it does the BB zeta. And even when we lower the ratios between effector and target cells, we saw that the tendency was very similar. They all have similar killing efficacy. And then to test the therapeutic potential of CD3 CARs, we implemented a stress test in which NALM-6 bearing mice was used. In this test, CAR T cells were given doses in which they start to fail. And remarkably, we could see that the BB delta here in pink, followed by BB epsilon in yellow and BB gamma in light green, significantly extended the life of the mice. And we have here zeta, in which the treatment was even worse than with the other CAR CD3. Then is to highlight that the BB delta show the best anti-tumor function. But then we also wanted to continue characterizing in vitro our constructs. So then we decide to analyze the calcium signaling capacity of our CD3 CARs, and we use a planar glass supported by a layer with different densities of the CD19. And it was found that BB zeta was the most sensitive CAR. It responded to the lowest antigen density. Meanwhile, BB epsilon also fluxed calcium, but only at higher antigen densities. Meanwhile, BB delta and BB gamma failed to flux calcium even at higher antigen densities. Consistent with this, we checked that BB delta and BB gamma CAR T cells fail to degranulate even in the presence of NALM-6 that are their target cells. Additionally, we wanted to check also the activation potential of these cells. And the analysis exhibited that it was the BB zeta CARs, the ones with the higher percentage of cells expressing CD25 and CD69 activation markers. At the same time, we also checked the release of cytokines. And it was again the BB zeta CAR, the one that produces the largest amount of cytokines, of the cytokines that we test. So interferon gamma, TNF alpha, and interleukin 2. It was also BB zeta CAR, the one that differentiates the most after the contact with target cells. it exhibited a lower proportion of naive cells if we compare with the other constructs, delta, gamma, and epsilon. In this construct, it seems that there were less differentiated populations that may be associated with the improved in vivo performance of these constructs. Later, in order to get an insight of what is happening at the transcriptional level of these CD3 CARs, we test the transcriptional programming of our CD3 CARs after antigen stimulation. CAR T cells were co-cultured with NAM6 cells for 24 hours and they're analyzed. And it was also reported stimulation dependent that was described before for the core stimulatory molecule that we have in our CAR. Therefore, we decide to include in this analysis a construct in which only 4-1BB was expressed. And the cells that were exposed to the antigen, we saw that in analysis, they were clustered by the CAR construct that they were expressed. And then when we check the transcripts of these BB cells that only express the core stimulatory domain, we saw that the CAR were the transcripts were differentially expressed if we compare with the cells that express our empty vector. So we then decide to use this BB construct as a reference to analyze the transcriptional changes induced by the individual CD3 intracellular domains. And then this analysis led us to see that BB zeta has the greatest impact on the transcriptome, followed by BB epsilon. A more moderate impact was performed by BB delta and BB gamma. And then when we compare the transcripts that were exclusively regulated by each of the intracellular domains, it was BB zeta, the one that uniquely regulates 49 genes, followed by epsilon, delta, and BB gamma only with 3 genes. In the overall pathway analysis of these transcriptomes, the construct activates signatures that are related to T cell activation, TCR signaling, and cell cycle. And now, in an attempt to mimic the persistent encounter of target and effector cells that occur in vivo, it was also designed a protocol in which CAR T cells were challenged several times with target cells. And at the end of this culture, the functionality of the cells that were re-challenged and non-re-challenged was checked. So, by means of cytometry of time of flight, we were able to see or to visualize the exhaustion landscape of the samples. The UMAP approach informs us the differences between each construct and the history of the antigen encounter. In the case of the non-challenged cells, we did not see major differences. In contrast, larger difference in the cellular proteome of the re-challenged cells was observed, and it was dependent on the construct that was expressed in these cells. Moreover, it was also observed that the cells that were re-challenged produced fewer pro-inflammatory cytokines. And later, we also confirmed these results by ELISA. Moreover, it was also shown that these re-challenged cells upregulate proteins that are related with exhaustion and dysfunction. For example, PD-1, TM3, and CTL4. These re-challenged cells also downregulate the T cell factor 1 that is expressed in a very peculiar subset of cells that have stem-like properties. This TCF1 correlates with lasting immunotherapies. And the proportion of cells expressing TCF1 when we saw each of the construct was in BB delta and BB gamma. And these results also are visible when we see and when we evaluate the killing ability of the CAR T cells after they are rechallenged. And it was BB delta, the construct that retained the best ability of killing cancer cells, followed by gamma, epsilon, and zeta construct. And with this, I would like to come in to summarize what we have done. So CAR containing CD3 gamma, delta, and epsilon were successfully generated and demonstrate to activate properly. This BB gamma, delta, and epsilon outperforming a preclinical model, in particular BB delta, by prolonging mice survival and reducing the disease progression. BB zeta and epsilon present a more potent activation compared to gamma and delta. And we also develop a protocol to test CAR T cell dysfunction and exhaustion. And after they re-challenge, they also secrete less cytokines, and it was only BB delta, the one which maintained the best killing capacity. The take-home message today would be that novel insights from the TCR were discovered and were visible through the creation of these BB gamma delta epsilon CAR T cells, and the harnessing of CAR T cells with the CD3 change diversity still has a high potential to improve CAR T cells. Then I would like to thank all the people that were hardly working in this project, starting with Dr. Misol Velasco, Simon Bandel, also Professor Dr. Susana Minguet, Professor Dr. Nathan Chanel, all our collaborators and people that contribute in this work. And for you.
Thank you very much, Valeria, for this super nice presentation. I don't know, so we will have a couple of minutes here of questions for you, and then join Q&A session at the end of both presentation anyway. So if you have any question, please do not hesitate to write them on the chat, just to start, or to raise your hand actually. Samuel, you raise your hand, so go for it.
Thank you. Very, very nice presentation. Really interesting. I was wondering, patients usually have previous treatment, and that might affect their T cells, and they might already have exhausted T cell before the leukapheresis and the collection of their T cell. What's your feeling about how your construct might changed? And you show that some were changing a lot and maybe not very efficient, the zeta part, and those who didn't change much the phenotype of the T cells were more efficient. So how can you reflect on what could happen on the patient?
Well, I think in that case, we maybe need to identify populations that still keep more a stem-like type to start a treatment, right? Otherwise, it will be, I think, very difficult to succeed with this treatment. But yeah, you're right. Maybe this would be a limitation. Yeah.
Thank you very much.
Rami, yes, please.
Yes. Valeria, thank you very much. Very nice presentation. So I was wondering, maybe I missed this part, the commercially available CAR T cells are which form, or are the mix of all these forms?
No. The commercially ones include only the zeta chain. This is the one that is used. That's why we thought that we can improve the properties using other chains.
And my other question quickly, so in the first slide, one of the first slides, you showed that all these forms had the same killing capacity in vitro.
Yes.
But then you moved to the in vivo and you saw some differences, and then you nail it down into more mechanistic profiling. So how do you explain that still in vitro, it's still the same capacity? Do you think that other cells are playing also a role by helping the CAR T cells to achieve its target? Or do you think it's really an intrinsic role of the CAR T cell?
Well, actually, I think this is a very interesting question. And I think it's more related with the constant encounter that it has, the T cell with the antigen. I mean, in vitro, the encounters are limited, right? Because at some point, the target cell, it's killed. However, in vivo, maybe you have more cells, and then the encounters with the antigens are more frequent, and that it leads to exhaustion. So the cells that will retain the ability to kill will be the ones that are less exhausted.
Makes sense. Thank you very much.
Thank you very much. So I think in the interest of time, for now, we should move on to the next speaker, and Crystal is going to introduce him. So we have a few questions, one question in the chat, but we can address it perhaps at the end of both presentations. Thank you very much.
Yes, thank you, Maria Astrid. Now I'm going to start by introducing our first speaker from EMEUNET, Dr. Samuel Bitoun. Samuel has a very interesting journey. He's actually a rheumatologist and an esteemed EMEUNET member, and currently working as an assistant professor in the rheumatology department of Professor Xavier Mariette at Bicêtre Hospital in Paris, France. He completed his PhD in immunology, supervised by Professor Mariette, and furthered his expertise with a postdoc at Claudia Mori's lab, University College of London. So not only in rheumatology, his academic journey also includes a fellowship in hematology, and he obtained an oncology degree. So his current research is focused on the link between cancer and autoimmunity, both in epidemiological studies and also translational basic science research. So welcome, Dr. Samuel.
Thank you very much for this kind introduction. I hope you can see my slide well in the correct format. And so I will, unfortunately, I don't have any research to present today on the topic, but hopefully soon we'll start working on the topic. And so I'm going to go over more clinical aspects and maybe some translational immunology. And first, I'm going to start about the CAR T-cell procedure. So how does it work? So basically, you take the lymphocytes from the patients. You will then transduct them with the construct that we saw already in the previous presentation, expand them, and then reinfuse them to the patient. And in order to make room for these T cells in the patient, people receive lymphodepleting chemotherapy, which is usually fludarabine and cyclophosphamide. In the cancer field, it's a major issue, this procedure, because it can take up to 6 to 10 weeks. So there's a risk of disease progression during the manufacturing. Also, you need to decrease the tumor burden of the patients before doing this procedure. So because there's a risk of inefficacy and also increased toxicity. And as I mentioned, the role of prior treatment, chemotherapy, steroids, and that's also true for autoimmune disease, might impact the quality of the T cells you will be collecting in this patient and might affect the efficacy you can obtain from transduced T cells. And then another major issue is the tolerance. So I'm going to go over the cytokine release syndrome. So it's really interesting thing that was highlighted by this procedure, and people, it can be very severe. So there's two main problems: the cytokine release syndrome and the ICANS, the immune effector cell-associated neurotoxicity syndrome, that can sometimes be life-threatening. They usually occur in the following few days of the patient. So in this period, the patient will be in the hospital after the infusion, and they're linked to the amount of T cells that are repopulating the circulation with the transduction, and they're also linked with several cytokines that we will go over with different phases of cytokines. What if you need to think about one cell that might be the key cell there? It's the macrophage. It's going to be stimulated both by the tumor debris and the T cell, and it's going to be able to secrete all these pro-inflammatory cytokines, induce vascular leakage and recruitment of more monocytes. I will differentiate into macrophage and lead to this fever and hypotension that characterizes the CRS, and that can lead to a need of ICU and intensive care unit for some patients. And so there's been a learning curve with these treatments, and now there's really clear guidelines to how to manage. So when there's only fever, no hypertension, no hypoxia, you just offer supportive care. But right when there is hypotension, so grade 2 with fever, if you exclude, of course, infection that can occur in this setting of recent chemotherapy infusion, you will administer tocilizumab, which is an anti-IL-6 receptor monoclonal antibody, And if this hypotension is still not responding, you will then transfer the patient to ICU and add dexamethasone for grade 3 and grade 4, and even higher doses of corticosteroids in grade 4. The ICANS is a neurological manifestation that are sometimes difficult to tackle with headache, seizure, aphasia, encephalopathy, confusion, or focal weaknesses. It's sometimes difficult. It can be only slight changes in confusion, and usually in young patients, which should not occur in this situation. The imaging is usually normal, and it's mainly done to exclude other causes of neurological symptoms like hemorrhage that can occur, of course, in the context of a hematological disease where thrombopenia is present. And so this ICANS, this neurological side effects of CAR T cells, there's also a central role of the macrophage and the microglia, which are the resident cells in the brain that will disrupt the blood-brain barrier and induce inflammation. And as you know, there might also be CAR T cell recruitment in the brain, even without tumoral infiltration and even without antigen there. And so the treatment of this side effects starts with the detection. So you need to use a specific questionnaire, which is called ICE, and you will quantify orientation, naming, commands following, writing, and attention. And then you will mainly use, according to this grading score, dexamethasone, and which is the main, so steroids, which will be the main treatment of the side effect that can lead to coma. So it can be serious. When you really go to the really severe and how to prevent and treat the refractory CRS, you might try to do pre-infusion risk mitigation, prevent severe toxicity, and one of the treatment that might be interesting if you're not tackling the severity, you can use anti-IL-1 anakinra, which is also a cytokine that might be important in these side effects. So there was a really good introduction on the constructs, and what we really have on the market, and the names are not very user-friendly. I don't know how they can come up with such difficult names to name those available CAR T cells on the market right now. And they mostly are either first or second generation CAR T cells with all the CD3 zeta and co-stimulatory with CD20N or 4-1BB or both, with different way to transduct the cells. And so the results right now really impressive and really become a standard of care in lymphoma, in large B-cell lymphoma. Here you can see that compared to standard of care, you really have a better event-free survival, a better now even overall survival with side effects. Cytopenia and cytokine release syndrome is really being tackled and managed really much better now, with only in this study only 6% of grade 3 CRS. And you see that some patients are cured of their cancer. Some people, you see the line is flat, the survival curve is flat for the yEFIS with the long follow-up. So that's really something interesting. When you compare the clinical effect of different constructs, you see that the CD28 co-stimulatory domain are interestingly more efficient. There were no head-to-head trials, but this is a French registry which is called Descartes. But this better efficacy is also linked with more CRS and more ICANS. So you can have both the better efficacy and the less side effects. And this is another field where CAR T cells have been trialed, which is multiple myeloma, so to target the plasmocytes using BCMA, not CD19 or CD20 on the tumor cells. And here it's much more disappointing, where you can see the survival, the PFS curves leads to relapse on all patients. No patients are cured of their myeloma, which was the hope with such a new and complicated and expensive treatment. And unfortunately, in myeloma, people are not getting cured with CAR T cells. And so the challenges are the price, of course, it's around 300 to 500,000 dollars or euros per patient because it's done by the pharmaceutical industry, and it's billed very, very expensively. So it's maybe interesting if you can cure the patient in lymphoma, but in myeloma it might not be. And there's really discussions in France for the reimbursement, and currently it's not reimbursed in France, CAR T cells for myeloma. And you have high acute toxicity, but also a delayed toxicity with cytopenia. And also, it leads to a prolonged immunodepression with infection. And here you can see that the non-relapse cause of death are mostly infections and not CRS or other or ICANS. So really, this is something important to take into account because you have a severe B cell or plasma cell depletion that will target non-tumoral B cells and plasma cells. And also this recent concern from the FDA, there is a 20 case of T cell malignancies that were reported to the FDA, which in last November led to the opening of an inquiry, and some of these T cell malignancies were CAR positive. Not all of them, but some of so the lymphoma may be due to the construct or the transfection of the construct. But what's good and what's really exciting in the field of rheumatology is that you can make your own. You can buy this machine for a couple 100,000 euros, and you can make your own in your own lab. This is a closed machine, so you take the T cell from the patient, and this will do everything and deliver you a pouch that you can reinfuse to the patient. Two different machines that do that, right? And this is what the opportunity that was taken by researchers in Germany, the group of Georges Schett in Erlangen, to bypass the pharmaceutical industry and to use it in autoimmune disease patients, especially in lupus patients, where it's been showing tremendous efficacy. So they homemade CAR T cells with 4-1BB constructs lead to very good improvements in clinical scores, great biological efficacy in all markers. It leads to B cell depletion and also to a kind of reset of the repopulation of the B cell after the effect of the CAR T wind. The repopulation will increase the naive B cells, as you can see here, and decrease memory B cell, activated memory B cell, and plasmablasts that are being shown to be deleterious in lupus. So really exciting, and which is really even more exciting is that for most of these patients, they are drug-free, in drug-free remission, and we are still waiting for reports that they actually relapse. So we'll see this in the future. There's a good tolerance, so only CRS in 3 out of 5 patients described in these patients with grade 1, 2 TOSI infusion, no ICANS, no infection in the study. And this has been expanded by the same group in several other diseases, like scleroderma, with the first patient published here with a mild, not very severe disease, because it can be, the procedure might be dangerous in this patient, but with mild pulmonary hypertension, lung, heart, kidney involvement, with nice efficacy and good tolerance in this first patient's report. It's also being extended in other autoimmune disease outside of the field of rheumatology, with the infusion in myasthenia gravis. Patient will also hear a nice efficacy on the clinical aspects. So how can we interpret this efficacy? How can we have deceiving B cell depletion therapy in lupus with rituximab, maybe hope with obinutuzumab, and so maybe targeting CD19 rather than CD20, where CD19 is expressed, you can see in more precursors of B cells in the bone marrow and later on the plasmablast and some short-lived plasma cells also, might induce a tolerogenic reset that may have an impact on on disease and have an efficacy. And so what are the challenges that are interesting in the rheumatology field? What is the role of the lymphodepleting chemotherapy? It's something you have to bear in mind, that there's also cyclophosphamide and fludarabine and significant doses that are administered to do the lymphodepletion. So what's the role in that? And I think it'd be interesting to look at a randomized trial where you do the lymphodepletion in the control arm to see if this has an impact on the disease. And also, interestingly, to the topic, the impact of lymphotoxic agent on the quality of the T cells you collect in the patient, and see how this might impact the effect. So in conclusion, the CD19 CAR T cells are really interesting therapeutic option, but maybe more in the refractory and severe autoimmune patients. So maybe lupus is a good thing, because in the refractory autoimmune patients early in the disease, you might not have such a severe organ involvement that might challenge the safety of the procedure compared to very severe scleroderma patients, for instance. Remember, it's quite expensive, but maybe we can reduce the cost by producing them in our lab next to the hospital. There's this risk, and really we need to follow this on the CAR T-cell lymphoma stuff, but I think it mostly overall opens the field of an intensive and new immunotherapy in autoimmunity that opened a window towards cure of this disease with maybe chimeric autoantigen T-cell therapy or bispecific or therapy, or even more advances that all come from most of them from the cancer field. And I think we really have a lot of things to learn from the people who work in cancer, and to make really advances in the field of autoimmunity. Thank you for your attention, and I'll be happy to answer some questions.
Thank you, Samuel, for this fantastic talk. I've seen that there's quite questions in the chat, but first, just one question as a clinician. Do you know if when you treat the patient with tocilizumab and when he had, a cytokine-induced syndrome, does this affect, the survival with the treatment of CAR T cell? I mean, if there is an impact of tocilizumab on the response with CAR T?
So that's been done, and it's really interesting question, because you can of course think that maybe diminishing inflammation might have an impact on the anti-tumor response, and this has been looked at actually in cancer, and especially in lymphoma, and it's been shown that people who receive the toci compared to those who don't have a worse impact on their anti-tumor efficacy of the CAR T cell. But really, that's something really that should be closely looked at. Also, maybe you can think that if you have a CRS or an ICANS, maybe it means that you have an immune system that's quite in good shape, that a similar thing has been done for cancer immunotherapy, where when you have side effects, it is thought that you have a better anti-tumor response. So maybe using when you use toci to treat immune checkpoint induced side effects, you don't see a reduced anti-tumor effect. So it might be that if you have a good immune system, you have a good opportunity to respond to immunotherapy of any kind.
Thank you, Sarah. So I have just a few questions, or short question and short answer, because I want also to have interaction between you and Anna Valeria. So first question, the person was quite confused about the delayed cytopenia. Is it a direct effect of the CAR T, or it is unexpectedly, for example, neutropenia? Do you know if anything like this?
Yes, actually, so the delayed cytopenia, the pathophysiology is under scrutiny right now, but it can occur in the following weeks and last for months. Thrombocytopenia sometimes, and neutropenia also, and anemia and it is thought that there can be, even if you don't detect the CAR T in the circulation, they can persist in the bone marrow, and there they can have a toxic effect on the progenitors. And that may be, and they're undergoing studies of a group in Gustave Roussy who's looking at ways to understand and treat these delayed cytopenia. So this hasn't been observed in lupus patients, but that's something that's not infrequent in cancer. And so if this happens in an autoimmune disease patient, might be a real challenge to tackle and to take care of.
And another very quick question: do you know if CAR-T there will be a clinical trial in children? I know that is also a topic on your center. So do you know anything about this?
Right now, there's none that is planned. The pharmaceutical company are starting the clinical trials in lupus, and they've reported an abstract in Spain from a Spanish team at the latest ACR. But currently, we're not aware of plans of that, but that might be something that's really interesting in Sjogren's, especially, of course, in severe patients.
There was just a paper in Nature this morning, so I checked. So I didn't see any clinical trial, but there's a phase 1 clinical trial for RA patients. So it's interesting also.
RA patients, that might be, that's,
Interesting.
I don't know if it's the best disease to...
The difficult to treat, I mean, the refractory patients, I think those one. Sorry, just other question. Is it only the duration of potential effect that people use CAR-T cells directed against? Sorry, I try to understand. So is it the only duration of potential effect that people use CAR-T cell direct against CD19 instead of anti-CD19 monoclonal antibodies in SLE or SSE? Sorry, I didn't catch really what the person means.
I don't know. I'm not aware of those studies of using the CD19 antibodies in lupus. I don't know of trials that have targeted CD19 specifically.
And do you know if the chemotherapy, I think pre-CAR-T are used in patients with autoimmune disease for pre-treatment, I mean?
Yes, of course. They have to use it, and they've used it. They've used the same regimen of fludarabine and cyclophosphamide.
Someone said that when rituximab was first introduced, many patients were drug-free for months to years. So yes, we have to check and watch if this patient, there are reboots or they're starting again
Yeah, that's true. But in the end, rituximab the clinical trial, the randomized trial didn't show any efficacy. So even though we're using it in lupus and other patients.
And in the chat, Anthony Saras share us a Nature Medicine overview. So if you want to have a look after that. So I want just to have a quick interaction between our two speakers. So my question, Samuel, you talk about autoantigen CAR-T. What's the interest power instead of alloantigen CAR-T? And what's the interest? What's the difference?
So what's really interesting, this is a slide about those. So basically, it's having a next generation and a new paradigm shift in autoimmune disease, and rather than killing, like in lymphoma, all the B cells with the tumor cells and the good cells who are responsible for fighting against disease infections, the idea is to target with these antigen recognizing. So you have an autoantigen on the CAR T cells that will recognize specifically autoreactive B cells, and this is something that's already been shown in mice ACR 2 years ago in antiphospholipid syndrome. So the idea is to kill specifically autoreactive B cells and to spare the other B cells who then be fully functional to fight against infection and to have a better tolerance of this disease, especially maybe also reducing CRS and ICANS by targeting a less amount of cells in patients. So this is how these antigen-bearing CAR T cells might be useful in the future.
Thanks, Samuel. So, Anna Valeria, do you know any idea how we can improve CAR-T, and apart from what you described, other subunits? Do you have any idea, or maybe will you have another project to follow? I mean, about what you describe in the paper. There's maybe another. What's the next step of your project?
Well, I think there can be several things. For example, we also work in CAR T cells targeting glycolipids or carbohydrates. This is something it's about to start getting more and more attention. And then maybe also the combination of these different CD3 subunits, right? Because in nature, the TCR have all of them, and they may have a very specific function. So maybe we can fine tune the the CAR T cells by including more than one CD3 subunit.
Okay, thanks. So I think we have two more questions. I think we have to close after this this webinar. So Anthony, you would like to ask a question?
Yes, thank you, Jean-Guillaume. So actually about the cytokine release syndrome, because I'm not sure if anyone had looked at the abstract, the latest ACR and EULAR, but actually it looks like cytokine release syndrome is not such a big problem when you use CAR T cells in autoimmune diseases. And probably this is explained by because in the lymphoproliferative syndromes, you have such a massive distraction of the B cell population that actually you don't really have that lymphoproliferation autoimmunity. So I think it was reported as a very, very rare complication compared to the lymphoma treatment. That's all just as a comment. But obviously we'll have more data in the following years as people and other centers use it more.
Thank you, Anthony. I think Samuel mentioned it that there was very well tolerated. Rami?
Yes,
So...
I have also a question for Valeria, which was also in the chat, regarding the latest update from the FDA on CAR T cells, toxicity, blah, blah, blah. Do you think that decreased T cell exhaustion might be associated with increased toxicity? Does it make any sense for you?
I mean, so far we have seen that the exhaustion lead to a reduction of cytokine release. So decreased exhaustion it will depend, right? So far we have seen that we will set release high amounts of cytokines, and even though the treatments are going on. So less exhausted cells, I think it will depend in many factors. So it's not only about the exhaustion, it will be also the target, the off-targeting, the activation, the intracellular domains that are involved in the CAR T cells. So I think there are many players in the game that can contribute or to a higher release of cytokines or decrease it.
Okay, thank you.
So I think I have to close this webinar because now it's 7 p.m. So I would like to thank our two speakers. That was a very fantastic talk, and we have great exchange on the chat and also with the speaker. And we'll also to thank all the moderators and all the persons that attended this webinar. Thank you.